When you receive Genosensor's EduPrimer DNA Profiling Kit, it will consist of two major components. First, a clear container containing temperature-stable components, and second, an icebox that contains temperature-sensitive components. The clear container contains all the components necessary for DNA collection and isolation. Solution A contains the necessary reagents for cell lysis and DNA extraction. Solution B contains a neutralizing agent that is used to prepare the sample for DNA isolation and PCR. Also included are cotton swabs that are used for collecting sample cells. Inside the icebox you'll find an ice pack used for keeping its contents cool. Beneath that you will find an aluminum bag that contains all the reagents necessary for PCR, including a PCR master mix, positive and negative controls, as well as a DNA ladder for gel electrophoresis. It is important that upon receiving the kit you place the aluminum bag in freezer storage at -20 degrees Celsius. Before you begin your experiment, you should prepare your heat block or water bath if a heat block is unavailable. Turn your heat block on and add water or other appropriate material to maximize heat transfer. Before using the heat block, be sure that the temperature sits stably at 95 degrees Celsius. Remove the PCR master mix from the aluminum bag and place out on ice to thaw. Once it has thawed, it is vital that before opening the tube you spin it down for 10 seconds, vortex it, then spin it down again. Be sure to keep the master mix on ice until it is ready to be used. Begin the experiment by adding 200 microliters of solution A to a labeled microcentrifuge tube. Then, using the provided cotton swab, swab the inside of your cheek for 10 seconds. Place the cotton swab inside the tube and cut it so that it will close tightly. Then, vortex the tube and place it on the prepared heat block for 5 minutes. Load the sample into a microcentrifuge and spin it down for 10 seconds to remove condensation. Afterward, remove the cotton swab from the tube using tweezers. Be sure to wash the tweezers with ethanol between samples to avoid contamination. Next, add 20 microliters of solution B to the tube to neutralize the mixture and prepare for DNA isolation. Vortex the tube to mix the solution, then place it in a microcentrifuge at 12,000 RPM for 1 minute. After spinning, a small pellet containing cellular debris may be visible at the bottom of the tube under the hinge. At this point, if you haven't already, prepare the master mix as explained earlier by spinning, vortexing, and spinning again. Begin PCR preparation by adding 10 microliters of the PCR master mix to a labeled PCR tube. Do the same for tubes labeled for positive and negative controls. Add 10 microliters of the sample supernatant to the appropriate PCR tube. Use the pipette to mix the sample in by pipetting up and down. Do the same with the positive and negative controls. If you do not immediately load the tubes into a thermocycler, be sure to store them on ice. Otherwise, load the tubes inside the thermocycler as shown here. After the initial denaturation cycle, have a cycle of denaturation, annealing, and elongation at 20 seconds each, repeated 40 times. Perform any other necessary setup, run the thermocycler, and unload after the time has passed. At this point, for classes with shorter time periods, you may unload the PCR samples and store them in a cool environment for a later lab period. Running the resulting PCR samples through gel electrophoresis follows standard procedures in a 1% agarose gel with a DNA dye such as Gel Red or CyberSafe. It is best to keep the gel in the dark during the cooling and gelation process in preparing the agarose gel, as well as when running the gel. Once the gel has run, view it under a UV lamp. For a full class, your gel should display the various genotypes found in the TPA-25 gene or the PV92 gene based on whether or not an elute insert is present in a given template gene. We hope this video has given you a clear idea of Genosensor's EduPrimer DNA Profiling Kit. You can find out more through our website at genosensorcorp.com.